Our next speaker is Miriam Meisler, who is going to talk about sodium channel mutations in epilepsy.
Thank you very much. Today I'm going to give you a little history of the back and forth that we've had with this very interesting gene, SCN8A, and end up telling you some of our more recent results. So SCN8A is a sodium channel gene, and I'll talk about two of the mouse mutants, our original mutant and one of our recent mutants, and also the human patients that have turned up with mutations in this gene. The first mouse led us to discovery of this sodium channel, which had not previously been recognized. And this mutant was caused by random insertion of a microinjected transgene, and it was lethal in homozygotes. And you can see here that this mouse is dragging his hind limbs. The hind limbs are paralyzed, and there's tremendous muscle atrophy due to lack of stimulation of the muscle by the nerve. And we were able to clone this gene, taking advantage of the fact that we knew what the transgene was that we had put into the mouse to begin with. So by forward genetics, we made a genomic library from the mutant DNA, screened it for the transgene sequences, and that took us to the flanking sequence, and we found that there was a 20 kb deletion in this gene. Now, SCN8A encodes a very large transmembrane protein. It's a neuronal sodium channel, and this channel is activated by the change of potential across the neuronal membrane, and it's the gatekeeper for initiating an action potential. So, as you'll see, if you lack this channel, you're deficient in firing, and if you have too much activity of this channel, you get hyperactive firing. And these large proteins have evolved for very rapid opening and closing. Those are the key parameters. Within 2 milliseconds, the channel closes, and if it fails to close, that causes big trouble. So the protein has 4 homologous domains. There's a positively charged transmembrane domain that's involved in opening the channel, and a specific pore and an inactivation gate that closes the channel. So since 1995, there have been 13 spontaneous allelic mutations in SCN8A in the mouse. Their locations are shown here. And they've been picked up because they cause very obvious movement disorders: ataxia, dystonia, tremor, paralysis. And it's been shown by people studying these mutants that many types of neurons are hypoactive. And this channel is really critical for initiating action potentials. Among these 13, 10 are complete loss of function, and 3 are partial loss of function. And based on these phenotypes, we undertook screening a couple of hundred human families for mutations in SCN8A. We looked at inherited ataxias, dystonias, essential tremor, without finding any mutations in this gene. We did find one family with a frameshift mutation, heterozygotes for a frameshift mutation, who had ataxia and also intellectual disability. And now we have more evidence that haploinsufficiency of SCN8A, I think, is associated with cognitive impairment. But the transition to human patients really came through human exome sequencing. So this was the first SCN8A mutation that led to the recognition that this is an important epilepsy gene. And this was based on whole genome sequencing of this pedigree by Michael Hammer in Arizona. So you can see the proband, his daughter, had early onset seizures and sudden death at the age of 15. And she had a de novo mutation. So looking at inherited pedigrees was looking in all the wrong places. And what we're finding in humans is de novo mutations. In this case, you can see it here in the chromatogram. And this caused a substitution at the end of the last transmembrane domain, a substitution of aspartate instead of asparagine. And if you look through the evolutionary comparisons, you can see this is an invariant residue in the reptiles, fish, and the invertebrate channel, as well as the other human paralogs. So there's reason to believe this would have a profound effect on the channel. Well, we carried out functional studies, And we found that this is, unlike the mouse mutations, these are gain-of-function mutations. And the problem is that the channel does not remain inactivated. And therefore, there's elevated persistent current, sodium flowing into the neurons, and therefore the neurons are more prone to generate a new action potential before they really should. And you can see that in these transfected cells. We're looking at how many action potentials are generated per second, and we've transfected either the wild type or the mutant channel. And at any of these stimuli, you get twice as many action potentials when you put in the mutant channel as if you put in wild type channel. So this is a neuronal hyperexcitability phenotype consistent with seizures. And since 2012, when we published that paper, there have been 150 patients identified with mutations in SCN8A. And the phenotype is a very severe early infantile epileptic encephalopathy, which has onset within the first year of life, very often even prenatally, with uncontrollable seizures. And all of these mutations are missense. There's no loss of function mutations. And all of them are de novo, arising in the affected child. And in these children, there's a 10% incidence of sudden death. We've done some functional testing of 11 of these mutations, and 9 of the 11 clearly showed aspects of hyperactivity in transfected cells. So why make a mouse model when you already all know this much? The human mutations are already identified. And these are some of the things we wanted to do by making a mouse model. We wanted to convince ourselves that this mutation was sufficient and nothing else in the patient background was contributing. We could learn a lot about which neurons are affected and where the source of these seizures might be, and as well as looking into the possible causes of sudden death and, of course, hopefully finding treatments. So we did talent targeting, and we obtained 5 out of 67 potential founders carrying this mutation. And here's some of the characterization of the mutant mice. The mice do recapitulate both the seizures and the sudden death. And here you see heterozygotes. These are the D+ animals, which have the patient mutation. And they start to have seizures and die at about 2 to 4 months of age. And interestingly, we only have 50% penetrance, even though these are back-crossed for 9 generations now. I don't think this is a genetic variant. I think because we've distributed the mice to several other labs, and the survival varies in different locations. So I think it's a question of how good the animal husbandry is, how long the animals will survive. These are hemizygotes. These are the mutant against a null, and they're more severely affected. So the wild type protein is offering some protection. And if you have a homozygote, it's extremely severe. So 2 copies is worse than 1 copy, even in the absence of a wild type allele. And here you can see the tonic-clonic seizures in all 3 genotypes. And because we could get homozygotes, we could look at the stability of the protein. And you can see on this Western blot, it's perfectly stable and present at the normal concentration. So we've looked at several types of neurons, and here you see spontaneous firing in cortical neurons, layer 2-3, compared with CA1 and CA3 in the hippocampus. And the wild type neurons don't exhibit any spontaneous firing during this 10-second recording. The cortical neurons in the mutant aren't firing, but both types of hippocampal neurons are firing spontaneously, suggesting, consistent with other evidence about this channel, that the hippocampus may be the source of seizures. Now, one thing we were able to do was to look at another organ. SCN8A is expressed at a very low level in cardiac myocytes and detected immunocytologically in the transverse tubules, where it's involved in, or perhaps involved in, excitation firing coupling. It's a very low level, and I was very skeptical as to whether this had any function or whether it was just this ectopic expression that you see with lots of tissue-specific promoters. But here you can see we looked at the function of the ventricular myocytes in culture. And this is just a measurement of the length of the myocyte. As you stimulate, you get a contraction. With each stimulus, you get a contraction in the ventricular myocytes from wild-type cells. But in the mutant, you have a very irregular excess firing pattern, similar to the excess firing of neurons. So this suggests that actually there may be a role for cardiac dysfunction in the sudden death in these patients. Now, drug testing, we were very interested. This is, of course, a very arduous process, but there was a drug in the literature that acted specifically on this persistent current of sodium channels, which we knew was affected in this mutant. So this is the structure of the drug, and we collaborated with Jennifer Carney at Northwestern, who had access to the drug in the form of food. So it's very easy to do a long-term experiment. And she fed the mice between the ages of 1.5 months before seizure onset and 6 months. And here are the untreated mice for comparison. And here are the mice on this diet. There was absolutely no loss during this period and no obvious seizures. And when she stopped the administration of the drug, the seizures began. So this was extremely encouraging to me as proof of principle that these patients might be treatable with drugs. And of course, it's a long way. This drug has never been in any kind of human program, but we're hoping to be able to move it there. And the last thing I want to show you is the genetic modifier approach. Here's another way to try to find a treatment. And in this case, we found that crossing with strain SJL rescues the survival of these mice. So we have a mutant mouse. We have them on two backgrounds. This one was on C3H, crossed with the wild-type SJL. Half of the F1s are heterozygotes. And if you look at the survival of the heterozygotes, among these 43 D-plus heterozygotes, we only lost one mouse, whereas the loss of the on the C3H background is shown here. And so, in principle, if we can find a single modifier, and we're starting to do the genetics with the help of the Jackson lab, we may find a new target for therapy. And J.C. Wagnon is working on this project. So, in general, this dynamic back and forth between human genetics and mouse genetics over a long period of time has given us a lot of information about the basic biology of SCN8A and its contribution to human disease. And as I don't have to tell this audience, phenotype-driven forward genetics, starting with an interesting mouse phenotype, has been very successful in the past for identifying the physiological role of human genes and is likely to contribute to understanding the other 50% of genes in the genome whose function is still unknown. Thank you very much.
Awesome, thank you.
I'm curious, in your genetic modifier screen, how many different strains did you test before you arrive at one that...
One.
So you were lucky.
Well, we put them on C3H, Black 6, and SJL.
Okay.
So this is probably a really, really naive question, but do traditional anti-epileptics work on those transformed cells?
No, that's the problem.
Yeah.
Have you looked at the heart, like without manipulating it? Like, can you do long-term telemetry?
Yes, we've done
something?
telemetry, and there are abnormalities.
Excellent. All right. Thanks for a great talk.
Okay, thank you.